Thank you very much for the kind invitation to speak. One of the reasons I accepted the invitation was to actually find out more about your Centre for Clinical Trials. I suspect that we have a lot in common, but I don't know. I may have got this completely wrong. But anyway, let's see. So just to tell you a little bit about the hub that Kay referred to. In 2008, the MRC, Medical Research Council, which is a government funding for medical research, the MRC made their biggest investment in methodology that they ever had done. And it's probably small beans when you look at NIH budget, but it was at that time it was £17 million. And they have subsequently, and that was for 5 years, and they've subsequently renewed several hubs for a further 5 years. So I am going to be in this position, fortunate position of having funding for methodology research for the next 5 years. The current network involves 6 hubs. You can see them dotted around the UK, including one in Northern Ireland. And this is the hub that I'm the director of in the Northwest, but I'm also, as the chair of this network, I'm also charged with getting these 6 groups to work together as appropriate, to give added value to some of the work that they're doing individually. Okay, so just to give you a flavour for what the hub network is charged to do, obviously the main reason for being is to undertake methodological research. That can be both led by us as individuals, but also we are commissioned to undertake pieces of methodological research, most commonly by the NIHR, National Institute of Health Research, which is the equivalent to NIH, but with a very reduced, much smaller budget. But they're the biggest funder of clinical trials, and they recognise, during their review processes, they recognise many methodological issues that they don't know the answers to, and they come to our network to commission pieces of research. But we also undertake training programs, so we have a cohort of PhD students, we run workshops, we offer methodological advice to other clinical trials units. So the hubs are not there to design fairly standard clinical trials, they're there to look at unusual designs and difficult issues. And we're also responsible for running the Clinical Trials Methodology Conference. So in the UK, we have had a bit of a gap when we haven't really had a methodology conference to go to. Some people have come to the SCT over here, but we decided, no, we need something for the UK community. So we've run this conference twice, and I'm really pleased to announce that we are going to run a joint conference with SCT in the Northwest in 2017. So it would be absolutely fantastic to see several of you there, and we make you very welcome. Okay, so that hub network is basically, this is the engine room of the hub network. We're organised, all the members are organised into one or more working groups. Hopefully, you might recognise some area that might be of interest to you. Some are very statistical or entirely statistical, adaptive designs, stratified medicine, evidence synthesis, and others not statistical, but extremely important for running clinical trials, recruitment issues, trial conduct, outcomes, etc. We have a new working group, which actually I lead, around health informatics and the potential, and I do use the word potential, the potential for health informatics to improve clinical trials. So our own individual hub is organised into 4 methodological themes, 2 of which are statistical, the early phase trials, many of which are adaptive designs, the efficacy and mechanism evaluation of targeted therapies. We're also one of only 2 hubs that has a theme specifically looking at patients' perspectives, and one of the things we're looking at the moment is when people talk about patient-centered trials, what do they mean? So can we define, can we categorize, can we improve people's understanding of what patient-centered trials are? I'm going to talk most about issues in later phase trials, so comparative effectiveness trials by and large. We have interests in particular areas of medicine, and I'll use one or two examples to illustrate the methodological issues. Most of what we do is actually generic across many different clinical areas, but occasionally, particularly in pediatrics, there are some issues that are specific to that area. So, one thing that we did a couple of years ago was in many clinical areas, groups get together and undertake priority setting exercises, and indeed in the Cochrane collaboration there's some work going on there. And we thought, well, why don't we do that for methodology? Why don't we see what we think the most important trials methodology questions are? So we did a survey of the clinical trials unit network in the UK. At the time, there were 48 clinical trials units in the UK running, so academic trials units. And we did a Delphi exercise, and I thought it would be quite interesting just to see whether or not the things that agitate clinical trials units in the UK, whether they're the same as the things that you battle with here. So there are no prizes, but I just wondered whether anybody could think what the top 3 or even the top 1 topic for methodological research. What was the biggest issue that was facing clinical trialists, clinical trials units? So recruitment, okay? Recruitment, well done. Number 2? Okay, I'll come back to that. Okay, number 3. So you've got 1 and 3. They're closely related, but 1 and 3. Anyone get number 2? No, we kind of link that to retention. Oh dear. So it's the reason I was here yesterday, outcomes. Choosing outcomes in clinical trials. So you don't need to look at this slide, but here are the top 3: methods to boost recruitment, choosing appropriate outcomes to measure, and methods to minimize attrition. So there is data monitoring on there somewhere, but actually methods for trial monitoring. Anyway, this is published, and we are quite interested to see whether these results are generalizable outside of the UK, not just in the US, but elsewhere. So I was just talking to Kay about that earlier. Okay, so what I'm going to do is actually, hopefully in the time I've got, give you examples of projects that we're involved in from each of those 3 topics. The third one is statistical. The first 2 aren't. So I hope I've got the flavour of this talk, the content of the talk right, and hopefully each of you will get something from part of it. So why is recruitment exercise in clinical trialists in the UK? So these are the results from the trial funders, the MRC, and the Health Technology Assessment Programme in terms of over the last 20 years, how they've been in terms of whether they recruited to target. 100% of the sample size that they wanted to achieve, did they? So there's an improvement from 10 to 20 years ago up to 5 to 10 years ago, and it has improved a little bit more. It's now at about 60%, but there's still massive room for improvement. Okay, so one of the things we wanted to look at, we run a lot of trials involving children with a national clinical trials unit for that area, and we wanted to see whether whenever we talk about trials involving children, there tends to be an a priori belief that they're harder to run. Okay, particularly pharmaceutical companies really think trials involving children are harder to run, and we wanted to kind of investigate that. And we wanted to look at barriers and facilitators to recruitment, whether you could achieve recruitment. So we have a lovely database in the UK of publicly funded and industry-funded studies that are running through the National Health Service. And we looked at that portfolio. There were only about 90 trials that had completed on that register. So we didn't have a big sample size, so we decided to limit the factors that we were going to investigate to 4. I had a clinical collaborator on this, and she felt really quite strongly. She had an a priori belief that the trials involving children, there would be a difference in the success of recruitment or not for trials that involved drugs compared to trials that were looking at some other intervention. She thought it would be much harder to recruit children to studies of drugs. I have my own interest in two methodological factors. I don't know if you're familiar, but there was quite a big study done. It was called the STEPS project. It was several years ago now, looking at factors that facilitated recruitment or not. They only found one significant factor, and that was whether or not a trial had a dedicated trial manager or trial coordinator. If it did, it was far more successful, far more likely to recruit. Interestingly, in the UK, there's been a really big increase in the number of academic clinical trials units. I was just hearing from Kay that it's perhaps the opposite here. But as I say, we now have 45 clinical trials units, which is a lot for the population size of the UK. And so we wanted to tease out whether it was just having a trial manager possibly not aligned to a trials unit, or whether actually there was something about being in a trials unit as well that helped the running of the trial. So here are the results. So in terms of whether or not it makes a difference in terms of recruiting to targets, to whether or not you're you're involved in a drug trial or not a lot of difference. Okay. We're also interested in whether or not it was easier to recruit in particular conditions. And as you can see, these healthy trials in the healthy population, they're by and large vaccine trials with a higher success rate than acute and chronic. So these are percentages of trials that recruited to target. In terms of the question I was particularly interested in, we looked at this factor of trial manager. So there were trials that had no dedicated trial manager, trials that did have a trial manager, but they weren't within a CTU, and those that were within a CTU. And you can see there is a benefit to having a trial manager, and it appears there's even more of a benefit if that trial manager is in a professional clinical trials unit. And that reached statistical significance. But interestingly, this is another topic that is hot. Yes, I'm sorry. Can you tell us what you mean by trial manager? Oh, okay, yes, good point. So trials have a chief investigator, a health professional, who by and large is the person that has come up with the idea. They have, in our unit, there are teams. There's a statistician, there's an information systems and data management person or persons, and then there's somebody called a trial manager who's responsible for obtaining the regulatory approvals that are needed, for monitoring recruitment at sites. Basically, they do the vast majority share of the work. They keep the trial ticking over. They maintain publicity about it, and they really help the chief investigator. So the chief investigator focuses on the clinical issues, pharmacovigilance or whatever. The trial manager sorts out all the operational issues. Is there a, sorry?
Are they mainly single center?
No. These are by and large multi-center.
All the centers?
Yes. Yeah.
So what's the range of
Of these trials, they range from probably 40 or 50 through to a couple of thousand. We're not talking about big mega trials of 10,000 or so. Some of the vaccine trials, they're the largest ones. Okay. But this was particularly of interest to me, this issue about whether or not you've done good preparatory work. Have you done a pilot study, a feasibility study beforehand? And as you can see, for those studies that had done some work first and presumably ironed out difficulties, they had a higher success rate of recruiting to target. Now, the reason we're particularly interested in this at the moment is because our main funder is very much in favor of internal pilot studies. So these were either internal or external pilots. So by internal pilot studies, what I mean by that is you apply for the funding for a main trial, but you put within it an internal pilot where, by and large, the main aim of that internal pilot is to review recruitment. And we are now getting into formal stop-go criteria, whereby the funder can effectively withdraw their funding if it looks like you're not going to be able to overcome recruitment barriers. So there's quite a lot of work being done on both the statistical implications of having stop-go criteria, but also the operational side of things. So that's that's work that's going on.
Another set of pilot trials where you do the trial to full stop and you apply for new funding and so on. So I guess your term is internal.
That would be external.
Yeah.
So that would be an external.
Are all of these internal?
No, they are a mix, but they are predominantly there's only 24 pilots. I think there was 20 that were external. Internal pilots are a new but increasing design in the UK. And I think it's because of the results that I showed you here that our funder is committing an awful lot of funding to trials that don't reach their target. And so they are more proactively managing the funding that they're giving. Okay, so that was a kind of high-level look at how we're doing. This is one area that most clinical trialists in the UK recognise as being one of the main reasons why we have such a poor success rate in reaching recruitment targets, and it's to do with the research governance and all the hurdles and bureaucracy that we have to go through. So we did a study in the UK. We had a very large epilepsy trial, SANAD 2, it's ongoing at the moment, where we were going to open 114 sites across the UK, and we wanted to look at the issue of how long it takes to get from ethical approval to opening the sites. Because anecdotally, in the UK, despite what the UK government may say, because they're trying to attract industry into the country, it's actually running at an average of, depending on the figures you look at, between 5 months and 8 months to open sites. And this has a detrimental effect on clinical trialists' enthusiasm. And in fact, if you wait that long, you may have another trial that's superseded the one that you're looking at. So we wanted to look at what are the factors that are prolonging this site opening, and is there anything that we're actually doing in the trials unit that is causing some of those delays? Maybe all our trial managers are going on holiday at the same time, and it's our fault. So what happens in the UK is the ethics committee for a long time, they were the hurdle. They were the place where the delays were happening, but we have got regulations now, which means you have to have the ethics committee has to give a decision within 60 days. Ethics is no longer the problem. They're not the cause of delay in the UK anymore. That's been sorted out. But we still have to get approval by each site's research and development office. And it's that is causing the block at the moment. So whilst there have been targets set by the UK government for once you apply to the site, they have to approve or not within 30 days, and they have to get the site open within 20 days, I think, or 30 days after that. So there are targets, and they're meeting them quite well. Actually, the reason that they're meeting them quite well is because they've moved all the delay and work beforehand. So this SSI submission doesn't really matter what it is. That's the time up to when the site is prepared to start the clock against which it's measured in terms of the UK government's targets. As you can see here, the median time from getting ethical approval for a site to actually submitting to that site to start its process is 7 months. I mean, it's just crazy. So we looked at the reasons for those delays. There were 4 main reasons why it took over 3 months. We figured that 3 months was a reasonable time to get things sorted. The main reason is something called excess treatment costs. What I'm not sure about is whether or not this is generalizable to the situation in the US. I think one of the issues here is that we have a system of the NHS, which on paper is very committed to clinical research, but actually in reality has difficulty actually funding some of the research. these two issues are all around sorting out site logistics. So we have to go and get pharmacy approval. The principal investigators at a site have to get all their team, their clinical team, to say it's okay, they're going to run this study. It takes an awful long time. And this we had to do for 114 different sites. We have recently heard in the last 2 weeks that the Health Research Authority in the UK is going to stop all of that, and there's going to be a single approval for a trial, and no site, if they want to participate, no site can go against that approval. So that is going to improve a lot of this. That's going to unblock this hurdle a bit. But this is an issue, and I don't know how it operates here, but ICH GCP says that you need to have documentation that anybody involved in a trial team is qualified, and we've interpreted that in the UK to mean that both the sites and the sponsor have to hold CVs. Don't know how you are, but in the UK, our clinicians don't tend to keep their CVs regularly up to date. This takes an awful long time to sort out. Plus, the sponsor and us as the trials unit who hold those CVs, we're not really qualified to look at those CVs and say whether somebody's competent. Their employer is, but we're not. So.
But isn't the bigger time problem the stuff up front from when somebody conceives of this trial and develops the data forms and all that other stuff before you get to this? Our experience is that if you're working with somebody who's starting a trial, there's going to be at least a year or a year and a half from the time that starts to having a data system form, IRB approvals and so on, much less to those things. Now, what's the upfront time?
Okay. So let's say in an ideal world, chief investigator comes to us with an idea 2 or 3 months before a funding deadline. Often it's 2 or 3 weeks, but 2 or 3 months. We'll run feasibility studies, possibly through the electronic health record or with the site. We'll run that up front. We make the funding application. HTA, the biggest funder, are pretty good. We can get a decision on that trial within about 4 months. So you're up to 7 months. Once we have the decision, the funders release money to allow us to work on the protocol to get what would be your IRB, our ethics committee approval, and our regulators approval. And we do that usually within 3 months. So within 10 months of a chief investigator coming to us, we should be open so that the clock is starting ticking. But then you get this.
I think Dr. Meyer was asking about form development and the manual procedures and so forth. Typically takes about a year.
Oh no. So when we know that the trial is funded and the funder releases the money to work on the protocol, we're always going to assume that everything's going to be okay, and we start that process in parallel of developing the forms and the database. Doesn't always work. There's sometimes a bit of a delay. But we have all our forms ready much earlier than all of the sites are ready to open. We always have everything ready when the first site is ready to open. Okay. So I've looked at the overarching picture. I've looked at the site level problems. Now I wanted to just show you the results, quite striking results from a piece of work that was done looking at the recruitment around the issue of recruitment when the clinician, health professional, discusses the trial with a participant. And in fact, this is pediatric studies. We did a piece of qualitative research. We took 4 pediatric studies. We observed, we had an observer and a tape recording of the consultation process where the clinical trial was being discussed. So this is with a doctor and a parent, typically, sometimes with a child who would be giving assent as well, depending on their age. The striking result is that this is typical of what the parents said when they were asked about, they've been approached about a trial. How did they feel about that? And generally, they were saying things like, well, doctors definitely should feel able to approach the parents. I definitely think that, because without approaching them, obviously they won't be able to do the trials. Some parents were actually excited to be asked to take part, for their children to be part of a research project. You don't want to think there's a trial that could improve your child's condition and your child hasn't been offered that. I would like to be asked. Well, they did understand when we checked their level of understanding. That seemed to be okay. So they want to be asked. They're not necessarily going to say yes. What was coming through clearly was that they wanted to be given the opportunity to make that decision. When you look at what the doctors were saying, almost uniformly, I think there were 40 interviewed, they were very nervous about approaching parents and children. So I will go and approach them, but I feel very uncomfortable doing it every single time. Each parent is different, causes me great anxiety, it's stressful for the family, it's stressful for you. So this was really in quite striking contrast. Parents wanted to be approached, doctors felt really nervous about approaching parents. So we're doing some work, around training programs for doctors in recruiting children into trials to try to give them more confidence that it's okay to do so. I'm not going to talk about several things that were already that were ongoing, but just in case any of you have interest in this area, and we can maybe pick it up this afternoon, I'm around. I talked about doing feasibility studies when the chief investigator approaches us and we want to know how many patients there are likely to be in this a certain condition. There's a big move and a big investment from the MRC in the electronic health record and using health informatics to improve many aspects of clinical research, but including trials. So I'm a bit of a skeptic, I have to say, that our electronic health record, whilst we have fantastic primary care records, at the moment, the access to that and the ease of understanding what's there and being able to sort out, yes, you can identify patients with epilepsy. You can identify children with asthma. But I don't know how your clinical trial protocols are. But when you start to try to identify the exclusion criteria, where typically everybody then disappears, you can't get that very easily from our routine health records. So at the moment, when we do feasibility studies, if we're really concerned, we run screening periods of time where we ask the sites to actually record how many patients are actually eligible according to the criteria in the protocol. So the trial hasn't started yet, hasn't even been funded yet. But we run those and we find that they're quite accurate. So our issue here is whether or not that there is a trade-off between the accuracy of what you can get from our electronic health records. It's quick, but it's not necessarily accurate, versus a screening study, which should be more accurate, but takes more time and is costly. So we're just doing an evaluation project around that at the moment. I won't go through the others. I think that was just something that Kay had mentioned was an interest here. Okay, so let me give you a quick introduction, a quick overview to what I was actually talking about all day yesterday. Not me talking, but I was in a meeting and Kay was there and we were talking about this. So I started my life as a statistician. I now consider myself to be a clinical trialist. Obviously not a clinical trialist, but this is really where I started. I was heavily involved in Cochrane systematic reviews and looking at these sort of what I call outcome matrices a lot and seeing the same sort of thing all of the time. These are the trials in a systematic review on the row, and these are the review outcomes. So the reviewer has chosen these outcomes to be important. And a tick means that outcome was measured and reported well, sufficiently to be included in a meta-analysis. Cross means nothing was mentioned at all about that outcome, and a circle means there was partial reporting. So there may have been a mean without standard deviation. There may have been the p-value was less than 0.05, but you've no idea what the effect size was. And the problem here is that there are lots of gaps. So wouldn't it be wonderful if these were all ticks? Okay, so much easier. The life of a statistician and a systematic review would be so much easier. The additional problem, two additional problems here. These crosses don't necessarily mean that outcome wasn't measured. There's overwhelming evidence, and Kay has looked at this as well, that there is reporting bias within trials as well as between trials. A problem now referred to as outcome reporting bias, although arguably it should be outcome non-reporting bias. And trialists are making decisions what to publish or what not to publish on the basis of the results, and therefore you're seeing a biased picture. So that's one additional problem to the waste issue in research, that it would have been so much better if, we had the same information from all of these trials. But the other issue is, who's deciding these are the outcomes of importance? At the moment, it's systematic reviewers. But should it be? Should it be systematic reviewers and clinical trialists and patients? So there are a couple of issues. So I started, as I say, as a statistician, trying to overcome the problem using statistical modelling, getting increasingly sophisticated by finding somebody I would call a proper statistician, Professor John Kopas, who's done a lot of work on selection mechanisms. And that's really quite a nice paper. And it works on the basis that if you have a clinical trial report in front of you, you're interested in a particular outcome, and there's nothing there about that outcome, then you make a judgment as to whether it's at high risk of bias, that you think it probably was measured, and the reason it wasn't reported was because it was significant. And there's a kind of list of criteria that you would, or questions that you would ask yourself to classify that report into whether it's high risk or low risk. And you use that in the model. But of course, any statistical model, you make assumptions. It's not the best approach. I prefer a prophylactic approach rather than that therapeutic approach. And one idea, it's not a new idea, Comet has not come up with this idea, of a core outcome set. This is an agreed, standardized set of outcomes that should be measured and reported as a minimum in all clinical trials in specific areas of health or healthcare. So what's important here is as a minimum. So this is not saying that these are core outcome set are the only outcomes that you should measure in a specific setting. You can record whatever else you like, but please measure the core set across all clinical trials in a particular area. Advantages of that are obvious. You get greater consistency. You get outcome matrices that hopefully the reviewer says core outcome sets are relevant to this systematic review. The outcome matrix is full of ticks. And you would have a reduction in selective reporting of those outcomes, not necessarily of the rest, but hopefully that would beget better practice. Okay, now this is an interesting one. We would hope that if you're developing core outcome sets that you're more likely to measure appropriate outcomes. Why do we think that? Okay, I'll come back to that point in a minute, but you should say, what's the evidence that core outcome sets are going to do anything? Are they going to work? This is the most well-known core outcome set. It's in rheumatoid arthritis. It was published around 1995. It was then recommended by the regulators in Europe and the States. And this is what happened. So in 2010, we're up to about 70% of trials measuring and reporting the core outcome set, which consists of 7 outcomes. I'll just flick through, that's not relevant. But you may say, well, that's just because everybody started to measure 30 or 40 outcomes and they just hit upon those 7. It wasn't anything on purpose. But what you can see, if you look at this blue line over here on this axis, is the mean number of clinical trial outcomes that were measured across these 300 trials. And it's stabilizing, 6.5 to 7. It's stabilizing. So there does appear to be, whether it's cause and effect, but there does appear to be a consistency being achieved in rheumatoid arthritis drug studies. So that gave us impetus to set up what's now what we call the Comet Initiative. And this is what we were talking about yesterday, about trying to bring together studies that have developed these core outcome sets, specifically for effectiveness trials, so the later phases of trials. And what we've done is set up a website And our key resource is this publicly available searchable database. So if you were interested in, so how have we populated that? That's a slide that's out of date. There's now over 500 references in there. We did a systematic review to find core outcome sets. It's a really hard thing to do because they're not indexed on Medline. We had to go through I didn't. Research assistants had to go through 25,000 abstracts. I looked at several of them. And there are 198 core outcome sets published. We know of over 50 that are ongoing. And this is showing the increase in interest in this area. 2013, that's just the first 4 months of 2013. So there's a real feeling amongst clinical research community that for too long, things have been in a bit of a mess. There hasn't been enough discussion across different countries, across different sites within countries about what are the important outcomes to measure. There is no gold standard for developing core outcome sets, but we decided we would at least write a paper on issues to consider. There are potential sources of bias in developing core outcome sets, the most obvious one being who are the stakeholders deciding what those core outcomes should be. It may be that there are particular interests from including a particular measurement instrument that somebody has developed and made their academic career on the back of. We have to be very clear about that. It's also true that there is a variety of different consensus methods that have been used, from sitting around a table and discussing and agreeing around a table through to a more formal Delphi online process. Lots of different methods. But what we're saying in this paper is that until we have a gold standard, we must at least be open and transparent in how this core outcome set's being developed. So there's just a note of caution. If you were to go away from here and say, well, I have an interest in obesity, so I'll have a look what's going on in obesity. We do not quality assure. We have not yet quality assured the entries in that database. We are developing a quality assessment tool, but we're just literally offering it for consideration by trialists so that you can at least see what has been done already. Okay. In terms of whether those core outcome sets include what we call, are they appropriate outcomes? Who should be deciding what the most important outcomes are to measure in all clinical trials given a particular scope? It's no surprise that all bar one of the ones we found involved clinicians. In COMET, we're very committed to the idea that patients have a key role to play in deciding what outcomes are important in effectiveness trials, and only 16% of those 200 involve patients. And worse than that, even when a study included patients, the percentage of people voting or giving their opinion for patients, that percentage ranged from 4% to 100. So there was one core outcome set that was entirely determined by patients. But generally, patients are really underrepresented in core outcome set work. Having said that, the majority of the ongoing core outcome sets involve patients. I think certainly in the UK, until about 5 years ago, it wasn't the dumb thing to involve patients in trial design. It most definitely is now. It's a requirement of almost all of the funding agencies. There are issues around whether it's tokenistic or not, but there is a requirement. So why do we think it's important to ask patients, and does it have any impact if you do? Well, that core outcome set for rheumatology, where I showed you the results, that was developed in 1995. There were 7 outcomes. It was developed by clinicians. No patients were asked. I'm not criticizing them. In 1995, I don't think anybody was asking patients what mattered to them. Or maybe good clinicians did, but clinical researchers didn't necessarily. In 2002, they started to ask patients, and in 2005, they'd reached consensus amongst the patients that actually fatigue was the most important outcome. And that wasn't one of those 7 in the core outcome set, but it will be in a revised core outcome set. And we're accumulating examples of where, when you ask patients, they don't necessarily say the same as clinicians. Okay, I'm going to skip over this, but there are there are difficulties or interesting methodological challenges in running consensus panels in this area. If you lump everybody together in a single Delphi exercise, possibly from different health professional disciplines, possibly patients as well, you have issues of what's the relative weight, the percentage of people that you should have from different disciplines. But you're also making a fundamental assumption that everybody in that single group is actually going to have the same underlying consensus. Now, rather than making that massive assumption, we prefer to run Delphi panels in disciplinary groups that we think are likely to have some underlying consensus. This was a project to develop a core outcome set in cleft lip and palate. It's the clinical teams are very multidisciplinary. We wouldn't expect them to necessarily have the same underlying consensus. This work is ongoing, but we also brought in information that parents and children had given us through online survey. I'm not going through online surveys. I'm not going through the results, but we will be publishing an interesting study that shows that when health professionals, they all give their own scores for a list of outcomes in round one. They're then shown the aggregated results by their specialist group. So cleft surgeons in round two saw what the group of cleft surgeons thought about an outcome. And they are then asked, do you want to change your mind about your score? Have a look at what your peers said. Do you want to change your mind? After we've been through that round, we then brought in the parents and children information, and we showed cleft surgeons both their results from round two, all the other groups' results from round two, and the parents and children's results. And we said, do you want to change your mind now? We found really striking results. All of these health professional groups changed their mind more when they saw each other's results and parents than they did when they saw their own peer group. But interestingly, and you'll have to wait for the paper, some of these groups changed their mind more than others. So I shall leave it to you to work that out. But there are methodological issues in this area. So that also then means there are potential sources of bias. So for anybody that's interested in the more qualitative side of this, there's a real research agenda that needs to be developed here, including how do you engage patients and the public in talking about outcomes, the language of outcomes. If you say outcome, it doesn't necessarily mean what we think when we're thinking about clinical trial outcomes. Okay. So COMET is having a good exposure at the moment. It's referred to, the COMET database is referred to in the new SPIRIT guidelines that give guidance for how you should, what you should include in protocols of clinical trials. It also has, it's a requirement in the UK for our biggest trial funder. You have to say that you've gone and looked at the COMET database, and you might say there's nothing there, or if there is something there and you don't want to use the core outcome set, you actually have to explain why. So it's gaining momentum, and the meeting yesterday that involved PCORI, NIH, AHRQ, Cochrane, represented by Kay, I think there was I think it's fair to say there was a great interest in understanding more about what's going on in COMET around the world to see whether or not maybe PCORI and NIH need to perhaps refer people to look at the COMET database. I wouldn't go so far as to say they might want to go as far as the UK funders, but hopefully there will be some consideration in the US as to whether or not this is a good initiative to get involved in. Okay. Just to finish on that point, so as I say, I must keep coming back to this. We are not quality assuring. We are just simply putting into the repository what people have done. My own feeling is there are some great pieces of work with some really good methodology in there, a wide range of appropriate stakeholders, and it's reported well. But then there's also some things that are not at that standard. So we are developing a quality assessment tool, a reporting guideline, and we're starting to develop a research agenda around the methods of developing core outcome sets. So I'll be very keen to hear of people. But importantly, as you can see, I am an enthusiast. And I have to be reined in, and we have an international advisory group who say, hang on, Paula, you have to really show that this approach works before raising everybody's expectations that it's all going to be all right. So we are working with clinicaltrials.gov on 4 case studies to actually have a look at monitoring the uptake from what they have in trial registers in 4 areas, the uptake of core outcome sets. Okay, so have I got 5 minutes? Okay, so the third thing, so I've talked about some work we've been doing in recruitment. I've talked about work we're doing on outcomes. If you remember, the third highest priority was methods to minimize attrition. Now, I'm not talking about methods to minimize attrition. I'm talking about statistical solutions to what you do when you have missing outcome data. I've got 2 examples. This is the motivating example. It's not actually to do with missing outcome data. But we ran what actually turns out to be the largest trial in anti-epileptic drugs, the SANAD trial, several years ago. I think it was over 10 years, and it was published in 2007 in The Lancet. And it compared a very standard anti-epileptic drug that had been around since the 50s, carbamazepine, with what was the new kid on the block at the time. But there's, a couple of new kids on the block every year, it seems, but against lamotrigine. And at the time, actually, there was a core outcome set recommended by the International League Against Epilepsy, and it was felt that the most important outcome to look at was the time to withdrawal of that randomized drug. I think that sounds like a rather odd primary outcome to have. The reason it was focused as the primary outcome was, by and large, it is the patient's decision when they come off an anti-epileptic drug. So these are newly diagnosed patients, which means they will have had 2 seizures or more. And the issue about anti-epileptic drugs is that obviously you're taking them to avoid having seizures. It may be that one particular drug doesn't control your seizures and you want to add in another, or you switch. Okay? But it could be that those anti-epileptic drug causes intolerable side effects. So there are 2 main reasons why you want to, there are two reasons for a patient deciding to come off a particular anti-epileptic drug, but they're making the trade-off. So they may have a rash from carbamazepine, but they're not having any seizures. And they decide they're prepared to make that trade-off and stay on carbamazepine. But at the same time, that rash may be intolerable. And they decide, I'd rather go on another anti-epileptic drug, run the risk that I might have a seizure on that other drug, but I can't stand this rash anymore. So I'm not particularly in favor of composite outcomes myself, but I can kind of see the merit in this. It is a patient-important outcome, but it is a competing risks situation. So these are the results for time to withdrawal of that drug for any reason, for either reason. Okay, and as you can see, because this is time to withdraw, you can see that, and it's a bad thing, okay, because you've either come off it because you're having seizures, or you've come off it because you're having side effects. So this is a Kaplan-Meier curve, and so when something is a bad reason, you want to be in the upper curve. So overall, lamotrigine seems to be significantly better. Patients are staying on lamotrigine longer. Now, when you actually look at the competing risks analysis of that data, you can look at the curve in a competing risk analysis for unacceptable adverse events and for inadequate seizure control. Look at the 2 things separately. So by and large, the seizure control is fairly equivalent, but you can see that the advantage of lamotrigine comes in terms of the reduced side effects. So this is the cumulative incidence. So in this curve, being on the lower line is better. The lower curve is better. So the benefit of lamotrigine comes from a reduced side effect profile. And when we published this in The Lancet, there was a letter written, outraged, because carbamazepine has been around for a very long time. Doctors are used to titrating it up. This is a drug where you start low and you titrate it up to a maintenance level. And the criticism that was leveled at SANAD 2 was that it's an open study, it's not blinded. It's very difficult to blind anti-epileptic drug studies that go on for a long time. It was an open study, and people felt that obviously carbamazepine had been titrated up too quickly because people were too used to what the side effects were going to be. And so it was biased against carbamazepine, and this was always bound to happen. Okay? And everybody had taken their time much more slowly with lamotrigine, and therefore, you weren't getting this side effect profile that comes from titrating up too quickly. So what do we do about it? We wanted to ask this. Sorry, this is what I've just said. Okay, we wanted to try and address this through an analysis. Okay. So the process by which we tried to address this was we wanted to try to take account of the titration rate of these two drugs. But they're different, they're given in different quantities. So there's both milligrams, okay, but they're not having the same pharmacokinetic effect. So what we needed to do first was to calibrate the dose that they were getting, and we decided to do that compared to the maintenance dose. So we had what they should have been on to stay on for years, and we had the titration rate up to that. And what we did was we set up a joint model. The survival time, the survival process is time to withdrawal of that drug. The longitudinal process is this calibrated dose. It's changing over time, and it's changing over time in relation to what's happening to the patient. So if you titrate up and you have a seizure, you will be titrated up higher to try to control that seizure. So it's in response to what's happening to the patient. Similarly, if you titrate up too quickly and you start to see a rash appearing, you may come down again to try to get rid of it. So there are two processes going on, and joint modeling is a means of trying to link those two processes by virtue of a latent variable or latent variables. And you have a standard longitudinal process. This is the latent variable here. And you have a standard survival process. In this case, because it's competing risks, we have cause-specific hazards. So this is a proportional hazards model that is specific to different causes. Those causes of failure being either side effects or poor seizure control. And again, you have a latent variable process in there. And what the assumption of the model is that conditional on these two latent processes, the longitudinal and the survival process are conditionally independent given these underlying latent processes. So what you do is you have to make some assumption about the relationship between those two latent processes. And actually, this is a fairly common assumption about proportional association. What's the parameter of interest here is this gamma parameter. If gamma is significantly different from 0, then there is clearly a relationship between those two latent processes, and there is some relationship between the longitudinal and the survival process. So when I show you the results, we're looking at the value of gamma to see if it's actually significantly different from 0. So I'll cut to the results. So this is the results from the standard competing risks analysis that doesn't take into account the the dose. Okay, so this is a hazard ratio for time to withdrawal for unacceptable adverse events. So here, a hazard ratio less than 1 is in favor of lamotrigine. It's saying that you are more likely to come off for unacceptable adverse events if you're on carbamazepine. It's less than 1, and it's significantly less than 1. For coming off for inadequate seizure control, as you can see, the best estimate is around 1, although that confidence interval is wide. When you take into account the titrated dose, what you see, first of all, looking at the gamma parameter, you can see here that it is significantly different from 0. There is a relationship, and it does suggest that the higher the dose, the more likely the side effect, which is as anticipated. Here, the side effect is, the gamma parameter is also significantly different from 0. And again, sorry, have I got that the right way around? Let me just, sorry, my brain's not quite there today. These are as you would anticipate for the association parameters, sorry. But what you could see here in terms of the hazard ratio, it's actually become slightly more in favor of lamotrigine. It's slightly lower, it's very similar, and they're very similar. So our conclusion from this analysis was, having taken into account the difference in titration rates, the evidence is still in favor of lamotrigine. And that was our response to the journal. One of the reasons that I wanted to show you this very quickly was to come back to this third priority of methods to minimize attrition. So if you're running a clinical trial where you have a longitudinal outcome, inevitably patients will drop out of that process. We had a study of acute episodes of asthma in accident and emergency units. We looked at nebulized magnesium sulfate compared to placebo. So it's at a certain step in the clinical pathway. We were measuring asthma symptom severity score, and you can see that these two curves are almost overlapping each other. And the question is, there was dropout, and are they overlapping because there is selective dropout? One of the reasons for dropping out, these patients are in A&E, and if their asthma symptom severity score, if they're getting better, the asthma symptom severity score will drop, and they will go home. And they may go home at an hour if they get better. They may go home at 2 hours, 3 hours. We were wanting to follow them up for 4 hours as a minimum. And it could well be that you're seeing this curve because of the dropout. If magnesium works, people are going home early, and what you're seeing is people who are not doing so well. Okay? So we wanted to have a look at this analysis of longitudinal data, taking account of potentially selective dropout. So under not taking into account the selective dropout, if you just looked at a complete case analysis, you would see that in a longitudinal model, the effect of magnesium versus placebo on the asthma symptom severity score is nothing. It's not significant, and it's the effect estimate is to drop the asthma symptom severity score by 0.1 at the most. It doesn't appear, and that's what you're seeing in that curve. Okay. But if you take into account selective dropout, potential for selective dropout through a joint modeling model. So here, in a joint model, you have a survival process, a time to event process, and you have the longitudinal process. So the longitudinal process is looking at the asthma symptom severity score as it was before, but this time you're kind of laying on a survival process running alongside where that time to event is time to drop out. For the majority of dropouts, we knew why. It was because they were getting better and they went home. For a few of them, we didn't know that. We thought that it was probably because the A&E team was somewhere else on an emergency, not measuring the asthma severity score for a patient. So potentially we were thinking that this was not likely to be informative. And the results from the gamma parameter quite clearly show that this is significantly associated with the asthma severity score. You are more likely to drop out as your asthma severity score goes down. That's as you'd expect. Here, there's no significant association between dropping out when you don't know the reason. The important point here is actually the comparison of magnesium and placebo having adjusted for selective dropout. What you can see here is, okay, it doesn't reach the magical significance level, if that's what you're after, but the defined clinically important level of a drop in asthma severity score here was, I think it was 0.2 on the score, and it's not quite reaching it here. So arguably, whether it's actually different from the results or not, it's not that clear. But this, at least in a different setting, may have given you rather different results clinically. So the important point here is that in terms of missing outcome data, again, you can use statistical methods, models, and adjustments like this, and you should use the best ones you can with appropriate assumptions. But by far, in my opinion, the best approach is to design the study well to avoid that in the first place. And that's where we need more methodological research. There is a systematic review in the Cochrane Library of methods to improve retention. There's very little randomized evidence of doing that. So to finish, and I apologize, I've gone a little over, okay, but to finish, we in the Hub Network, we're in a very privileged position to have this methodological funding, but we want to collaborate and offer that funding for appropriate projects outside of our network. We can pump prime projects. The Comet initiative started with £50,000 from the Hub Network, and we think it's done well, and it's been good value for money. It's generated a further £1 million in grant income, so that was from small beans, really. And we have PhD students, and we're always looking for opportunities for them to come and meet with other PhD students elsewhere. And so we have training, travel grants, and we're very keen when we run training and research workshops to not just be insular in the UK. We actually want to look at issues that are affecting trials elsewhere. So if anything I've said has chimed with problems in trials in your centre, it would be really good to potentially think if there's anything we can do collaboratively. Thank you. I apologise that it's taken a while. Yeah.
You spoke about Comet and the core guidelines for outcomes. And do you have any suggestions for related diseases? So one of the things that I know conference is the blood pressure trials that report hypertension as one of their outcomes for blood pressure trials. This diabetes has so many different possible diseases that stem from diabetes that could not be.
So, sorry. One of the most difficult but most important things that you need to do when you're thinking about core outcome sets is to define the scope of the core outcome set. And there's a sort of parallel, I think, within Cochrane reviews as to are you a splitter or a lumper? So do you define a core outcome set that is going to address all trials of any intervention in breast cancer? Or do you define a core outcome set that is going to address all trials of chemotherapy in breast cancer? Do another core outcome set that's all trials of radiotherapy? And so I can't give you a specific answer to that clinical area. And as with Cochrane, there's a mix. People have different approaches. I don't know what the right answer is, and I don't know what the right answer is here. One might imagine that if you looked at trials of breast cancer chemotherapy, trials in breast cancer radiotherapy, I'd be surprised if there wasn't some things in common, probably the benefits outcomes, the harm outcomes will probably be different. Does that answer your question? Okay.
Yeah, I imagine this is related. When I heard you discuss the core outcomes that it seems that the easy part is saying the outcome, and hard part is the there because I'm thinking among the person with blood pressure. And so people can measure blood pressure through that, and they can almost believe that they need to get several different days worth of what the variance, or 3 days worth, getting the random zero, and it's not really relevant unless it's collected at 6 months, after you've had an intervention at that point. So it seems to me that it's easy to say blood pressure, so it's easy to say fatigue, but doing it right in a way that people really, it almost gets you back to some critical, other trial.
So we promote, we like a two-stage process. The first stage being what should we be measuring, and the second stage being how, but linked to how there are other levels, including when. But the reason we do it that way, and COMET really is focused on what, in that COMET database there's a mix. Of those 200 core outcome sets, 100 of them recommend what, and leave it at that, and 100 recommend what and how. So they've done, I'm not sure how many go on and look at when but the reason we start with what is because if you start from how and you say, I'm only going to measure things for which I have an instrument that is psychometrically valid, you would never have measured fatigue. You wouldn't have, there isn't. So to get some structure, I do like the hierarchy that Deborah Zarin published in the New England Journal, which includes the level of aggregation, the time point. But I guess we're trying to start somewhere and not try and bite off all difficulties with standardization. Does that?
I agree with you, but I think the injustice of what by itself, that's not enough.
Yeah.
You cannot stop there.
Yeah. I think I totally agree. I absolutely agree. It's a long process. And I guess methodologically, not methodologically, I guess logistically, operationally, some people have found it easier to do the what. And then there's a whole piece of work to be done around how, and Promise are involved in that, and Cosmin, and I think the stakeholders might be different as well. The people who decide what is important may not be the same people who decide how. I've been asked as a statistician to get involved in a core outcome set project on the what. I don't know anything about the disease, but I might go into a process looking at how. I haven't done psychometric stuff for a while, but it's not obvious that the same group of people should do both. Not to me, anyway.
Sorry.